Hello, everyone, and thank you for taking the time to join us today for this exciting webinar being brought to you by Scintica Instrumentation. My name is Tanya Coulthard, and I will be your host for today's webinar. Our webinar today is entitled GSK3 Inhibition and Muscle Wasting Disorders, including Muscular Dystrophy and Spaceflight. Today's webinar will be given by Dr. Val Fajardo, a Canadian Research Chair in Tissue Remodeling and Plasticity at Brock University here in Ontario, Canada. Dr. Fajardo's research focuses on muscle wasting conditions as well as muscle metabolism with the goal of developing clinically relevant therapies as well as on the discovery and characterization of calcium regulatory proteins. During today's webinar, Val will talk about his recent work focused on the role of the glycogen synthase kinase 3, also known as GSK3, and its role in muscle wasting conditions such as muscular dystrophy, as well as in spaceflight. Specifically, looking at the inhibition of the enzyme in the treatment and management of these pathologies. Additionally, Val will explore the effects these inhibitors have on cardiac function in these disease models. Before we begin, I'd like to briefly mention a few housekeeping items. We anticipate that today's presentation will be close to 45 to 50 minutes. This should leave us some time at the end to answer your questions. Please submit the questions in the Q&A dialog box throughout the presentation, and I'll work with Val to get through as many as we can during the hour that we have together. We will create a transcript of the questions we do answer during the session, as well as those that we may not have a chance to address at the end. And we will distribute this over to you in the next few days. Please also note that we are recording today's session, so if for some reason you lose connection or you can't hear everything clearly, you will have access to the video file for your own review later. So now, without further ado, I'd like to turn things over to Val to take on the presentation today. Val, you're muted.
Okay, everyone can hear me okay?
We can. Thanks, Val.
Thank you for the introduction. And I'm not gonna lie, the countdown there almost seemed fitting for this presentation, again on muscular dystrophy and then on spaceflight. I'm gonna spend most of my time, admittingly, on this talk, specifically on the muscular dystrophy side of things, because we have a lot of data. It's actually unpublished data, and I'm happy to share with you today because we do believe that it will be published soon. But again, thank you for the introduction, and thank you everyone for being here today. So I'd like to start off my presentation, but I don't see it moving forward. Here we go. We're okay there. By acknowledging the students and the people who have contributed their time, their effort to producing the data that I'm going to show you today. Again, some of it will be published data, a lot of it will be unpublished. And so starting on the left side here, we have Kennedy Whitley, who is a master's student in my lab, and she's actually looking to defend in a couple of weeks. She, along with Sophie Hampshire, who's a PhD student in my lab, comprise the muscular dystrophy team. They've worked together on separate items and separate projects to produce the data that I'm going to show you today. And I should acknowledge this fellow over here, Sebastian Silvera. If you've watched any of my presentations for Scintica before, you would know that Sebastian was an all-star in my lab, really helped enable a lot of the things that we're working on. And unfortunately for us, he's moved on to another position, but he has contributed tremendously towards the data that I'm going to show you today. And then over on the right here, we have Ryan Baranowski, a master's student, a first-year master's student in my lab, and then Jessica Braun, who's a PhD student in my lab. And by the way, Sophie and Jessica are both accelerated PhD students, so they're both top-notch PhD students in my lab. I just thought I'd point that out. But Ryan and Jessica, they quote unquote represent my spaceflight crew, I guess, in the muscle plasticity and health and disease lab. So these are the people that have worked on the experiments or conducting the experiments and the data that I'm going to show you. So today's talk, I've set it up to go over three main things. And the first thing is GSK3 or glycogen synthase kinase 3. And what I want you to take home is what I want you to know is why we are interested in it. And there are four main reasons why we're interested in GSK3, specifically inhibiting GSK3, as Tanya mentioned. And those four main reasons will serve as the building blocks for my talk today. The second thing I'm going to cover today, of course, is GSK3 inhibition and Duchenne muscular dystrophy. So I'm going to focus a lot on skeletal muscle and cardiac muscle, but I would also be remiss not to highlight some of the recent work from my colleague, Dr. Rebecca McPherson, who focuses on cognitive decline. So I've only set aside 2 slides for this tangent. So I have a 2-slide tangent on the cognitive side of things with Duchenne muscular dystrophy. And I think it's really important to highlight. And then finally, I'll finish off with GSK3 and spaceflight. And I'm hoping to show you why we think it's a potential therapeutic target for mitigating the muscle loss and weakness that occurs with spaceflight. And that, again, is GSK3 inhibition. So moving along here, so GSK3 stands for glycogen synthase kinase 3. And as its name implies, it regulates glycogen synthase. This is the enzyme that's responsible for producing glycogen. Now, this GSK3 was first identified in muscle to regulate glycogen synthase. And the way it regulates it, if you're familiar with kinases essentially add phosphates to their substrate. And so when glycogen synthase kinase 3 phosphorylates glycogen synthase, it inactivates glycogen synthase, prevents glycogen production. But aside from glycogen synthase, we now know that GSK3, glycogen synthase kinase 3, has over 100 substrates. So it's a serine threonine kinase that has over 100 substrates. And in muscle, there are 2 isoforms, GSK3 alpha and GSK3 beta. Now I have GSK3 beta in bold fonts and underlined font here because it is the dominant isoform found in muscle. So a lot of the data that you'll see today will be focusing on GSK3 beta. And over on the right here is a simple schematic of how GSK3 functions. So again, it's a kinase, and it phosphorylates its substrate. So let's imagine this substrate here labeled S. You'll see that there's this P circle here, meaning that it's phosphorylated. And glycogen synthase kinase 3 likes to have a primed substrate, meaning that it's pre-phosphorylated. And this allows for recognition of this substrate. And eventually what GSK3 does is it utilizes ATP, which is the cellular currency of energy. And it uses that ATP to phosphorylate this substrate. So now by the time GSK3 is done with it, this substrate has 2 phosphates in this case. So again, it's a kinase, meaning it adds a phosphate. So why are we interested in GSK3? And as promised, there are 4 main reasons why we look at GSK3 and specifically at inhibiting this enzyme. And that's because inhibiting this enzyme can, 1, increase muscle size and mass. And I'll take you back to a couple of early studies in the early 2000s by Dr. David Glass's lab and Dr. Frank Booth's lab. And both of them showing that when you inhibit this enzyme, when you stop glycogen synthase kinase 3 from working in muscle cells, in this case, they're growing muscle cells in a dish. When you stop this GSK3, you actually see that the muscle cells in the dish grow to be larger. Their myotube diameter is larger. So in the case here, published in Nature Cell Biology, what they did is they took C2, C12 muscle cells and they transfected this dominant negative GSK3 beta mutant variant, essentially meaning that this GSK3 is inactive. And you could see that control versus the negative or inactive GSK3, the myotubes are quite large in comparison. Over here from Frank Booth's lab, you see the same thing, but in this case, they inhibited GSK3 with insulin-like growth factor 1, which is a well-known anabolic factor. We know that IGF-1 will increase muscle size. And what we see from their experiments is that that leads to an increase in serine 9 phosphorylation of GSK3. So this is important. This serine 9 GSK3 phosphorylation is important because it's inhibitory. Remember, GSK3 likes to recognize a primed substrate. When GSK3 is phosphorylated, it almost acts as this pseudo substrate. So it doesn't allow GSK3 to recognize any other substrates except for, I guess, itself. So this phosphorylation is inhibitory. Now that's important to know because I'll be talking about this phosphorylation status quite a bit in my talk. So it's best we sort this out now. And if you look at insulin-like growth factor 1, and you look at myotube area, again, you're inhibiting GSK3, you see a significant increase in myotube area, just like with the inactive variant of GSK3 beta. The authors also took the cells, the C2, C12 cells, and treated them with something called lithium chloride. Now, lithium is a well-known inhibitor of GSK3. I don't have their data. I'm not showing you their data here, but this lithium would also produce the same effect of increasing inhibitory serine phosphorylation. So a well-known GSK3 inhibitor. And again, you see that lithium chloride treatment increases myotube area. Now, I will admit that this dose or this result, while it proves the concept, this dose is quite high. It's supraphysiological. And so my lab reproduced these findings. Actually, Nigel Kurgan, who's a PhD candidate, soon to be completing his PhD with Dr. Noda Klencher here at Brock University. He completed an independent study in my lab. And what he did is he treated C2, C12 cells with a much lower dose, and it's 0.5 millimolar, again, compared to the 10 millimolar that I just showed you. Now, this 0.5 millimolar dose is reflective of what we see in the clinic. For those of you who know about lithium, you probably know it for its treatment of bipolar disorder. And 0.5 millimolar to 1 millimolar is actually that therapeutic range. So we wanted to go on the low end of this therapeutic range of lithium and treat the muscle cells. And lo and behold, we do see an increase in phosphorylated GSK3 beta. We also see that in the alpha isoform as well. So it's inhibiting both isoforms, which is good. And then if I can point your attention to this panel A here, and this is our immunofluorescence staining technique to look at myoblast fusion. So the blue dots here are actually nuclei, and the green tubes are the muscle myotubes. And you can see with lithium treatment, there's a lot more myotubes being formed, and they're bigger in size. And we see that the nuclei inside these myotubes, there's a lot more in them. And that's what we call myoblast fusion. So we see a significant increase in fusion index. Now that's important for not only growth, but also for muscle repair. Another thing that Nigel noticed is that we do see increases in myogenin and myosin heavy chain. Now these are markers of muscle differentiation. So not only are we seeing larger myotubes, we're seeing, as you'd expect, more signs of muscle differentiation. So it enhances muscle size and myogenic differentiation. So again, just to recap, we're interested in inhibiting GSK3 because it can increase muscle size and mass. And I'm not going to get into it today because I think it's beyond the scope of today's talk. But there are multiple mechanisms by which this can happen. GSK3 inhibition has been shown to increase protein synthesis while reducing protein breakdown. And for muscle growth, it's all about balance. We want to, in terms of simple mathematics, we want to increase protein synthesis while reducing protein breakdown so that we have a net gain in muscle protein and therefore muscle size and mass. And so GSK3 inhibition can increase protein synthesis while reducing protein breakdown. It actually represses the expression of E3 ligases that are important for the ubiquitin proteasomal degradation pathway. And as Nigel showed you, GSK3 inhibition can actually improve myoblast fusion and myonuclear accretion. So muscle cells are multinucleated. And there's a theory out there, although there is quite there is some debate to this, suggesting that if you increase the amount of nuclei surrounding a fiber, that can help support muscle growth as well. So again, there's various mechanisms to which inhibiting GSK3 can increase muscle size and mass, but nonetheless it does. And we are really interested in it for that reason. And another reason is that it can increase muscle strength. And so this is Kennedy Whitley, again, a master's student in my lab looking to defend in the next couple of weeks. And this is a project, it's a side project, so above and beyond her thesis project. So thank you, Kennedy, for this. And what she did is she took 3 to 6 month old male C57 BL6 mice, and we fed them with a low dose of lithium chloride in the drinking water. We just provide it in their water bottles, so it's really easy to do, and at a dose of 10 milligrams per kilogram per day for 6 weeks. Now, when we measure the circulating lithium concentration, we see that it's calculated to 0.02 millimolar. Again, remember the common dose with bipolar disorder is 0.5 millimolar. So for the treatment of bipolar disorder. So we're much lower than that. And despite being much lower, we still see significant increases in force production in both the soleus, which is a postural muscle, and I'll talk a lot about that, especially with spaceflight, and then the extensor digitorum longus, EDL, which is a fast twitch muscle. So we see here the controls are the open circles, so they produce force as we increase the frequency of stimulation. So the rate with which we zap the muscle, I guess, or electrically stimulate the muscle, you can see that you can increase specific force production. However, when we treat the mice with lithium, we do see a significant increase both in the soleus and in the EDL. So it enhances muscle strength. The third reason is that it promotes the oxidative fiber type and fatigue resistance. Okay, and this is important to us, especially for Duchenne muscular dystrophy and spaceflight, as you'll see later on in today's talk. So this is a immunofluorescent staining protocol that we run in my lab. Actually, these are some images from Sebastian. So thank you, Sebastian. And what I want to say with this slide is that muscles are comprised of a mix of different fiber types. So we have these type 1 fiber types that are blue, these green 2A fiber types, these black fiber types that just look unstained, that's why they're black, and then these 2B red fibers as well. And aside from color, these fibers can be differentiated by other things, not just their color. So this is a slide I use quite a bit in my teachings, but also in my presentations. And these fiber types can be differentiated by various contractile and metabolic properties. So if we look at the contractile kinetics and metabolism, or the way in which they produce ATP, and their fatigability, whether or not they are most or least susceptible to fatigue. If we put the fibers in a spectrum from top to bottom, your blue type 1, your 2A fibers in green, your 2X fibers that are unstained and therefore appear as black, and your 2B fibers that are red, you could see that in terms of contractile kinetics, the way with which, the speed with which they contract and relax, the type 1 fibers are the slowest, whereas the 2B fibers are the fastest. So it goes down in that spectrum. When we look at metabolism, and again, we're talking about ATP production, how do they produce ATP? We see that the type 1 and 2A fibers are most oxidative, and that just means that they use oxidative phosphorylation, and most of their ATP producing sources come from mitochondria, whereas the 2X and 2B are more glycolytic, so they are using cytosolic sources or enzymes to produce ATP. And this is related to their fatigability, because the type 1 and 2A fibers use the mitochondria, which are able to produce more ATP, albeit at a slower rate. They are least fatigable, whereas the 2X and 2B fibers are most fatigable. So again, your type 1 and 2A, your type 1 is your slowest, most oxidative, and least fatigable fiber. And then your 2B is your fastest, most glycolytic, and most fatigable fiber. And then I have here that calcineurin signaling will promote the oxidative fiber type by activating NFAT, something called nuclear factor of activated T cells. And I haven't introduced calcineurin yet, but I will now. And so calcineurin is a very important molecule or enzyme, that we look at in my lab. And it's a calcium-dependent phosphatase. And so that's opposite of GSK3. GSK3 is a kinase, right? So it adds a phosphate, adds a P onto its substrate. Calcineurin actually does the opposite. It's a phosphatase, so it will take that phosphate away from something. And that something, again, is NFAT, or nuclear factor of activated T cells. And when calcineurin takes that P away from NFAT, that allows NFAT to enter the nucleus. Okay, this transcription factor can enter the nucleus, and along with other factors and enhancers, what NFAT can do once it's in the nucleus is actually increase the expression of oxidative genes, or genes associated with the oxidative myogenic program. So calcineurin promotes the slow oxidative phenotype. Again, it's a phosphatase. It takes that phosphate away, whereas GSK3 puts that phosphate back on. So GSK3 actually represses calcineurin signaling, where calcineurin takes that phosphate away, GSK3 adds that phosphate back. And when GSK3 adds that phosphate back, well, NFAT can no longer enter the nucleus. It has to exit the nucleus, and we don't get this increase in the oxidative myogenic program. So the points that I have here, GSK3 represses calcineurin and NFAT signaling, and inhibition of GSK3 should therefore promote the oxidative fiber type. So you've got to think about inhibiting the inhibitor. If you get rid of this, or you reduce its activation, then inherently you amplify this side of things. That's the idea. Okay, so if you inhibit this, you amplify this, and therefore increase the oxidative myogenic program. And Kennedy, using that same lithium feeding study published in the same paper, showed just this. So in the soleus, we have this increase in inhibitory phosphorylation of GSK3. Remember, increased phosphorylation of GSK3 specifically at that serine site means that we have inactivated it. Because of that, we have less phosphorylation of NFAT, which is good because it can enter the nucleus and then promote the oxidative myogenic program. And therefore we have an increase in myosin heavy chain 1. Those are those blue fibers, the type 1 blue fibers. So we get a significant increase in that with lithium treatment. Again, these are the closed bars versus the open bars. And then when we look at the fatigability of these muscles, when we isolate the soleus muscle and we mount it into a contractile system provided to us by Aurora Scientific, and what we do is we stimulate the muscle every 2 seconds at 70 hertz for 5 minutes. So just imagine zapping the muscle every 2 seconds to produce force. You would imagine that as you are doing that consistently, that force would drop, okay? And that's what we call fatigue. What we notice is that when we treat the mice with lithium and we look at their soleus muscles and their ability to fatigue, we see that they maintain a higher percent of initial force throughout that protocol, leading to a significant increase in the area under the curve. So we're promoting the oxidative phenotype that uses more oxidative metabolism, and going along with this, we see an improvement in fatigue resistance. Now, the last reason, the fourth reason why we're interested in GSK3, again, why we're interested in inhibiting GSK3, is that we think it can improve calcium regulation, and specifically the Scintica pump, or the sarcoendoplasmic reticulum calcium ATPase. And I'm hoping this doesn't lag on your side. If everything's okay and working in order, then you should see 2 videos. The first video, thank you to Aurora Scientific for this video. It's a video of a muscle bundle contracting and relaxing. And over on the right here is this video that I made, oh gosh, back when I was a PhD, trying to depict what happens when a muscle contracts and relaxes. So if it's working out on your end and it's not lagging, then you should see that the timing should be spot on. And so when we want to contract our muscles, we get this signal from the motor neuron that initiates what we call an action potential. And this action potential essentially leads to depolarization of the sarcolemmal membrane, which then travels down the transverse tubule. So this depolarization is just a change in voltage. And this change in voltage or depolarization is sensed by these green proteins here, the dihydropyridine receptors. And you can see these loops, these black loops that I've drawn, that means that they're connected with these blue squares, which represent the ryanodine receptors. So when these green dihydropyridine receptors sense this change in membrane potential or voltage, what happens is the ryanodine receptors will open up and they'll release these red dots, which are calcium. So the red calcium gets released out into the cytoplasm, and that activates the myofilaments. So in order for us to relax, when this muscle bundle relaxes, that calcium signal actually needs to be taken back into this reservoir, which we call the sarcoplasmic reticulum. And to bring that calcium back into the SR, or these red dots back into this reservoir, we need the action of the Scintica pumps. And the Scintica pumps are here. So the Scintica pumps will transport these red dots, these calcium ions, back into the sarcoplasmic reticulum using the energy harness from ATP hydrolysis, because it's what we call active transport. And what we think GSK3 inhibition can provide is we think that GSK3 can improve the function of these Scintica pumps. And Sophie Hamstra, a PhD student, soon to be candidate, she's completing her comprehensive exams next week. So that should be PhD C soon. She showed this in my lab, actually, with her undergraduate thesis. And what I'm showing you here is a Scintica activity PCA curve. So on the x-axis, we have PCA. It's the negative logarithm of calcium concentration, just like pH. So it's a little bit backwards, but what I want you to know is that as you move from left to right on the x-axis, you're increasing calcium concentration. And what we're measuring is Scintica activity, or its ability to hydrolyze ATP. As you increase the calcium concentration that Scintica is exposed to, you increase Scintica activity, because Scintica is a calcium-dependent enzyme. But what we notice is when you treat mice with lithium, that same low dose of lithium that Kennedy Whitley used, and this is, by the way, is in the left ventricle, so in the heart. When you treat the mice with lithium, this red curve here, we notice that there's a leftward shift. And this leftward shift means that it doesn't take that much calcium to activate the Scintica pump. So we've increased Scintica's apparent affinity for calcium. So that's an improvement in Scintica function. And we think that this is due to GSK3 inhibition and its role in increasing Scintica protein. Okay, so we get a significant increase in Scintica, and we get a significant reduction in phospholamban, or PLN. Now, PLN is this regulator that binds to Scintica. So it physically binds to Scintica. And what it does is it inhibits Scintica function. It actually reduces its affinity for calcium. So when we treat with a GSK3 inhibitor, such as lithium chloride, we improve Scintica function, in part because of this improvement or increase in Scintica protein. We also see this in MDX mice. And I'm going to be talking a lot about the MDX mice. So again, we see this leftward shift. You're looking at the red dash curve here. These are the MDX mice. And again, looking at left ventricles, the MDX mice treated with lithium, that same low dose of lithium, again, for 6 weeks. So 10 milligrams per kilogram per day. Now, this MDX mouse model is the preclinical mouse model of Duchenne muscular dystrophy. So I think that's a good transition into my next section of my talk. But before that, again, I'll just recap why we're interested in GSK3. Inhibiting this enzyme in muscle can increase muscle size and mass, increase muscle strength, increase or promote the oxidative fiber type and fatigue resistance, and therefore improve calcium regulation. And the bottom line of here that I've got written down is that all of these factors listed above are really important in conditions of muscle wasting, especially muscular dystrophy and spaceflight. So again, these are the building blocks. I'll be talking about each of these as I progress through the slides for muscular dystrophy and then eventually spaceflight. So moving on to GSK3 inhibition and Duchenne muscular dystrophy, again, knowing that I have 2 slides to go off on a tangent here, looking at cognitive decline. So this is my, I guess my 3-minute thesis slide on Duchenne muscular dystrophy to introduce the disorder. So it's this severe muscle wasting disorder. So you've got this cartoon image depicting a very healthy muscle. When you look at the dystrophic muscle, you can already tell that that is going to be weaker. There's a lot more space between light pink staining. That's what we call fibrosis. There's a lot more of these blue dots or purple dots in this muscle. And that's immune cells coming in to try to repair damage, but eventually adding to the damage. So this muscle looks damaged and weak. And so Duchenne muscular dystrophy is a severe muscle wasting disorder. And the second point I have here is that it's an X-linked recessive disorder caused by X-linked recessive mutations to the dystrophin gene. So essentially what that means is it primarily affects boys because they only have one X chromosome. And there are over 7,000 mutations in the dystrophin gene that can lead to Duchenne muscular dystrophy. And these mutations in the dystrophin gene, they all lead essentially to one thing, and that's the complete absence of dystrophin. So dystrophin's not there. And so what's dystrophin? So let's point your attention here to quadrant 3. And dystrophin is a rod-like protein that provides structural stability. So it's this purple rod-like protein here. And this protein, what it does essentially is it connects the muscle, the extracellular matrix above, to the cytoskeletal network below. So it provides structural stability. So as our muscles contract and relax and contract and relax, there's a lot of wear and tear, as you can imagine, with contracting and relaxing and contracting and relaxing, the push and pull, especially at the membrane level. With dystrophin there, it ensures that our membranes don't tear. However, in patients living with DMD, with the complete absence of dystrophin, so dystrophin is not there to provide that structural support, along with other proteins in what we call the dystrophin glycoprotein complex. When dystrophin is not there, as our muscles contract and relax, we are more susceptible, or they are more susceptible to membrane tearing. And this membrane tearing is what leads to necrosis and apoptosis and muscle degeneration. And so this disease is a progressive disorder, and it leads to early mortality. So early on in the childhood stages, there's dysfunctional ambulation. Teenagers are often wheelchair bound, but the disease quickly progresses into cardiorespiratory failure. So that leads to premature death. Unfortunately, these patients will only live until into their 3rd or 4th decade of life. And with no cure for this disease, the main goal in terms of management is to slow the progression, because we know it is a progressive disorder. Can we slow the progression? Can we prolong the lifespan? But can we also prolong health span? Not only can we increase the number of years, can we increase the number of healthy years? And so that's the research objective that we have in my lab, and that's to slow the disease progression. Again, through GSK3 inhibition, because it can increase muscle mass. This is a muscle wasting disease. It can increase muscle strength. It's a muscle weakness disease. It can increase oxidative phenotype, that the type 1 blue and type 2A green fibers. This will become more important as I explain it in the next 2 slides. But it can also enhance calcium regulation. Again, those are the building blocks for my talk. So in my lab, we use something called the D2 MDX mouse, and it's a more severe model of Duchenne muscular dystrophy. So the MDX mice were originally identified in the mid-1980s. And these MDX mice, like patients living with DMD, don't have dystrophin, so they don't have that structural stability. Now, these BL10 MDX mice were the original MDX mice. So they're backcrossed onto a C57 BL10 strain. The problem with the BL10 MDX mice, and this is well established, the limitation with the BL10 MDX mice is that in terms of disease severity, it's really mild. So it's not as representative or not as translatable to human DMD, where the disease severity is quite high. Recently, researchers have started using what we call the D2MDX mouse, which is the MDX mouse backcrossed onto a DBA2J background. So here's some results from this publication in the FASB journal. And these are from 10-week-old mice. So this is a really young mouse. And what you can see here are H&E images across quadriceps, gastrocnemius, tibialis anterior, triceps, and diaphragm. And what you're looking for with these H&E images in terms of damage are these areas occupied by blue dots, the immune cell infiltration, so a lot more inflammation happening, these light pink staining, so these are fibrotic areas, even here as well. And you can see a lot more of these abnormalities in the D2MDX mice, again, at a young age. So showing in terms of histopathology, the D2MDX mice are a lot worse off compared to the BL10 MDX mice, and meaning that it might be more clinically representative of the disease. And when you look at muscle strength is important. In this case, they're looking at the EDL, the extensor digitorum longus, a fast twitch muscle. From A through B through C, this was published in 2016, you see that the EDL muscles are smaller in the D2MDX mice, the upside down gray triangles, okay? And they produce the least amount of absolute force and also specific force. So the force being normalized to the size of the muscle. So they're smaller and they produce less force. So the D2MDX mice is a more severe model with more histopathological abnormalities, and they are weaker. Kennedy, as part of her actual master's thesis, took these muscles from BL10 MDX mice, so the less severe model, and D2MDX mice, and wanted to characterize what happens to GSK3 expression, but also activation. So looking at that phosphorylation status. And what she found is really quite interesting. She found that the D2 mice, whether or not you're wild type or MDX, shown here in red, have more total GSK3 content. And when you look at the phosphorylation status of that GSK3 is actually less phosphorylated in these D2 mice. So the key point, I have it written down here in the bottom right corner, is that the D2 strain have more GSK3 in their muscles, and the GSK3 that's there is actually more active because there's less of that inhibitory phosphorylation. So we wondered whether or not GSK3 is then contributing to this worsening of disease severity in the D2 MDX mice compared with the BL10 MDX mice. And so to get at this, we treated the D2 MDX mice with tideglusib. And it's a potent non-ATP competitive GSK3 inhibitor. So that's just the mechanism by which it inhibits GSK3. It's not really that important for today's talk. I think what's most important is that it's the most clinically advanced GSK3 inhibitor. It's in clinical trials for Alzheimer's disease, diabetes, and myotonic dystrophy, which, by the way, is another form of muscular dystrophy with its own pathology and pathogenesis. So it's completely separate from Duchenne muscular dystrophy. But this was important to us, this factor here, because we wanted to test a drug that could potentially lead to the rapid clinical integration of the drug to patients living with DMD. So that's why we chose tideglusib. And again, this is part of Kennedy's thesis, but I also have Sebastian, who is our former lab technician, and he really enabled this project. We provided tideglusib at 10 milligrams per kilogram per day. This is a clinically relevant dose. We tried to make it clinically relevant, and it was done through oral gavage 5 days a week, and the oral gavage was performed by Sebastian, so thank you, Sebastian. And the treatment lasted for 2 to 4 weeks, depending on the measures that we were going to conduct. But we started the treatment at 6 to 7 weeks of age, so at a young age in these mice. And this is a result here over on the right after 4 weeks of treatment. And the first thing I want to show you is that GSK3 content is reduced. So remember, these D2 mice have higher GSK3, and tideglusib treatment actually reduced GSK3. And although it didn't change the phosphostatus, when you normalize the phosphorylation status relative to total GSK3, we see an increase in phosphorylation. And again, this is inhibitory phosphorylation. So there's less GSK3 that's there, and the GSK3 that's there should, in theory, be less active. So this was quite successful in our eyes. And so the first thing that we wanted to look at again was muscle mass, the first reason, so tideglusib and MDX muscle mass. And here we have EDL muscle mass relative to body mass. Okay, and the wild types are in the open bars. The MDX vehicle, not really treated with anything, are the closed bars, and the tideglusib are the red bars. And what you could see is that, as you'd expect with the D2 MDX mice, they have smaller muscles. Okay, but 4 weeks of treatment restored this to wild type levels, which we're quite happy about. And we use the Insight small animal DEXA at the beginning and end of treatment. It's really quite easy to integrate this small animal DEXA in terms of longitudinal studies. So we measured their lean mass at baseline, and then we also measured them post, so 4 weeks of treatment. And what you can see, again, consistent with GSK3's role in regulating muscle mass, when you inhibit GSK3, the tideglusib treated MDX mice had the largest amounts of gain in lean mass compared to both wild type and the MDX vehicle treated mice. So we're quite happy about that. What about muscle strength? Now, there's a few ways that we can look at muscle strength. And the first way, it's the same color coding here. Wild types are open, MDX vehicle are closed, and then MDX tideglusib are red. The first way is to look at their ability to hang on a wire. So we place the mice on a wire, and we time their ability to hang on that wire before falling off down to a safe bedding area. And we normalize that to body mass. And so that's why it's called impulse. And we see that the wild type mice compared with the MDX vehicle are better able at hanging on that wire. So this is a measure of not only force production, but in my mind, also a little bit of fatigue resistance. And so we see this significant reduction in the MDX vehicle. But 4 weeks of treatment improved their ability to hang on this wire to the point where it's not significantly different from wild type. So that's good. The other thing that we looked at was we took EDL muscles, we isolated them and mounted them onto the contractile apparatus that we have from Aurora Scientific. And again, as you increase the frequency of stimulation, the rate with which you electrically stimulate the muscle, you will increase force production, because that's what we call force summation. And you see this nicely in the wild type mice. You don't see this as nice in the MDX vehicle mice. However, tideglusib treatment actually improved specific force production, specifically at tetanic stimulations and also submaximal stimulation, so 100 hertz and 150 hertz. So we've improved muscle strength and also fatigue resistance. And we can show this again when we take that muscle and we run that fatigue protocol. If you remember from Kennedy Whitley's slide, every 2 seconds, we stim the muscle 70 hertz for 5 minutes. And in this case, what we've done is we've tracked the amount of time it takes to reach 50% of initial force. So if your muscle is more susceptible to fatigue, then it will take less time to reach 50% of force. And that's what you see with the MDX vehicle compared to wild type. However, the MDX tideglusib took a little bit longer, meaning that we've improved fatigue resistance, again, to the point where it's not different from wild type. And this improvement in fatigue resistance, maybe you can guess where I'm going with next, this improvement in fatigue resistance is associated with a promotion of the oxidative fiber type using that immunofluorescent fiber type staining technique. And what we see, although it's not significantly or statistically different in terms of the type 1 and type 2A individual fibers, we do see an increase, but it's not less than 0.05. When we combine the oxidative fibers, type 1 and 2A, and compare them in terms of the proportion of the oxidative fibers versus glycolytic fibers, the 2X and 2B, we see that the tideglusib treatment, again, this is in EDL, increased the amount of the oxidative fibers, both type 1 and 2A together, and reduced the proportion of the 2X and 2B fibers. So we've inhibited GSK3 and we promoted that oxidative fiber type. This is important, okay, because I've touched on this. These fibers can be differentiated based on contractile kinetics. Do they contract slow? Do they relax slow? Their metabolism, do they use mitochondria or cytosolic productions of ATP? And their fatigability, are they least or most susceptible? But we can apply another spectrum on these fibers, and I'll point your attention to this bottom left corner here. And that is the fast glycolytic fibers are most susceptible to muscular dystrophy. So if we look at type 1, 2A, 2X, and 2B from top to bottom, and we look at the dystrophic pathology, the type 1 and 2A fibers are least susceptible to dystrophic pathology, whereas the fast glycolytic fibers are most susceptible. And there's a variety of reasons that can explain this. One of the reasons that I really like is the expression of utrophin. So this was published by Shakalako and colleagues in 2003 in PNAS, showing that utrophin is much higher in type 1 and 2A fibers. So utrophin is much higher in these blue and green fibers. Now utrophin probably sounds to you a lot like dystrophin, and as it should. So dystrophin is this rod-like protein. Utrophin is also this rod-like protein that provides structural stability. So utrophin sounds like dystrophin, it looks like dystrophin, and in fact can function like dystrophin, especially in its absence. So in DMD patients that don't have dystrophin, if you can increase utrophin to compensate for this, that will provide additional membrane stability. And again, utrophin is more abundant in the oxidative fibers. So when we've increased these oxidative fibers with GSK3 inhibition, it was no surprise to us that we see an increase in utrophin mRNA. And then when we look at the H&E images and look at the histopathology, so this is the MDX vehicle EDL, you see the light pink staining for the fibrosis, the blue dots for immune cell infiltration, so more inflammation. You see that a lot with the vehicle and almost restored with the tideglusib treatment. This is actually 2 weeks of treatment here, these H&E images. We've also shown this 4 weeks as well. And when we look at necrotic area, which is the areas occupied by the light pink staining and blue dots infiltrating, we see significant reduction in the tideglusib treated EDL. One last marker of muscle damage is serum creatine kinase. So creatine kinase is a really important enzyme in terms of muscle energetics. So we like to keep that in our muscles. However, when our muscles are damaged, we release that creatine kinase out into circulation. So we can use that therefore as a marker of damage. And so the MDX vehicle, same color coding here, has significantly higher creatine kinase in their serum, indicative of muscle damage. 4 weeks of treatment reduces this significantly, not to the point of wild type, but significant from MDX vehicle. So again, we've attenuated some of the muscle damage, which is really good for us. Okay, the last reason, right, why we are interested in inhibiting GSK3 is calcium regulation. And I'm going to focus here on cardiac calcium regulation, the left ventricle. And this is some of Sophie Hamsh's PhD work. And what I'm showing you here is a calcium uptake assay. Remember, the circuit pumps will bring calcium into the SR reservoir. And so when we provide ATP to the muscle, we can see calcium goes down as it enters the SR. This happens nicely in the wild type, but definitely not as nice in either MDX treated protocols. But we do see significant reduction in the starting calcium. So wild type versus MDX vehicle have more starting calcium, and this is blunted after tideglusib treatment. And that tells us actually that perhaps we've mitigated the calcium overload that happens in the MDX muscle. So some subtle improvements in SERCA function. And when we look at that SERCA activity PCA curve, right, you're increasing calcium concentration from left to right. You see that tideglusib treated here in the red have greater SERCA activity across submaximal and maximal frequency. So we have improved SERCA function with GSK3 inhibition in the hearts. And to give you some functional context here, we have some high frequency ultrasound data using the Scintica Prospect T1. So we've just purchased this infrastructure here, and I'll be quite honest and admit that this technique is a new technique in our lab. So we are quite green with this technique. So I do have to thank our host, Tanya, who really supported us and spent 2 or 3 days with us, helping us collect valuable, reliable, and usable data. So really kudos to the folks at Scintica for that, helping us collect this viable data. And I don't think Tanya has seen this data, so this is probably the first time she's seen this or heard me talk about it. But here we go. So we looked at stroke volume and cardiac output, and long story short, we didn't see any differences. But that's not really surprising. That's actually consistent with Coley and colleagues in 2016. We actually don't see cardiac contractile abnormalities until later on, about 28 weeks of age. So this is 9 to 10 weeks of age. But no one's looked at the diastolic function side of things. And so what I'm showing you here is isovolumetric relaxation time. And you could see that the MDX vehicle take longer to relax, which is perhaps indicative of diastolic dysfunction and should make sense when you think about SERCA dysfunction, because it's important for relaxation. And then when you improve SERCA function with tideglusib treatment and GSK3 inhibition, you restore that diastolic function. So this tells us that perhaps diastolic dysfunction and SERCA dysfunction will precede cardiac contractile abnormalities, and maybe targeting SERCA function and diastolic function will help mitigate that cardiac contractile abnormalities. Okay, so those are the 4 reasons again why we are interested in GSK3 inhibition in the MDX mice. And a lot of the work that I've showed you today is looking at the short-term treatment, so 2 to 4 weeks. So in terms of future work, we want to look at long-term tideglusib treatment, and we want to answer whether it's well tolerated, is it more effective, and can it prevent cardiac dysfunction or future cardiac dysfunction and prolong lifespan and health span of these mice. And here I'll go now on that 2-slide tangent. And one of the things we're interested in collaboration with Dr. Rebecca McPherson is whether or not this long-term tideglusib treatment will prevent cognitive decline. And so who is Dr. Rebecca McPherson? She is a researcher, associate professor at Brock, and her research focuses on what happens to brain cells with Alzheimer's disease. Now, why is that important? We know that there are cognitive impairments often reported in patients with DMD. However, the mechanisms remain unknown. And I'm happy to say, as part of this collaboration that was just recently published a couple of weeks ago, I can read the title here, Characterization of Alzheimer's Disease-like Neuropathology in the DBA2J MDX mouse. So we saw signs of Alzheimer's-like pathology, enhanced amyloidogenesis. I'm not going to touch on it or expand on it because obviously I'm not the right person to talk about it. But it's interesting because when you go on PubMed or Medline and you type in GSK3 and Alzheimer's disease, you're going to find 1,600 results. And that's because GSK3 has long been implicated. Its overactivation has long been implicated in Alzheimer's disease. In fact, we are constantly developing new and potent GSK3 inhibitors to try to prevent or combat Alzheimer's disease. So one of the future items that we have on our list is if we're using tideglusib in the long term to improve muscle function, well, let's see if it also improves cognitive function. Okay, that's my 2-slide tangent. I'll move now to Spaceflight because I know I'm probably going overboard. So I'll quickly go through the GSK3 inhibition in Spaceflight and explain why we think it's a potential therapeutic target. Okay, and in Spaceflight, the goal that we have now is human deep space exploration to Mars and beyond. However, spaceflight, though really exciting, comes with several physiological risks. And that includes fluid shifts, muscle and bone loss, which we are particularly interested in. There's also psychological risks that ultimately will reduce the success of the mission at hand. So we need to mitigate these risks or slow down these risks. And so the muscle loss that we experience in space operates on the premise of if you don't use it, you lose it. Okay, so you've got a healthy muscle. If you go up in space, we actually consider it to be a rapid aging model. You can see signs of sarcopenia. And we see this mostly in the postural muscles, like the soleus. So the soleus I've got shown here helps you stand up and fight that never-ending downward pull of gravity. So throughout the day, it's more active compared to its fast glycolytic, compared to other fast glycolytic muscles. So when you go up in space and you're exposed to microgravity, it's these muscles that are most affected that will experience the atrophy. And because of this atrophy, astronauts are often told to exercise roughly 2.5 hours per day, but that includes setup and breakdown. And we know that this exercise, though beneficial, is not quite sufficient in combating the atrophy and the remodeling that occurs. We actually see in the soleus, this is actually in mice, 91 days aboard the International Space Station, there's a slow to fast fiber type shift. So they become more fast glycolytic and fatigue susceptible. You start to see these red fibers starting to pop up. Again, those red fibers are your fast glycolytic 2B fibers. And of course, because of the muscle atrophy, we do see muscle weakness. However, when I was presenting at a conference with a colleague of mine, Carlo Reggiani, he brought up this point where that muscle weakness that we see with spaceflight or any form of disuse or unloading cannot be merely due to a reduction in muscle size. I won't get into it, but there's got to be other pathways in play, and he suspects that calcium dysregulation, and I'm going to show you some data telling you why we're also on board with that. So what can GSK3 inhibition offer? I'll be really quick with this because a colleague of mine, Boris Schenkman, wrote this really good review and published it recently, the role of GSK3 in the regulation of protein turnover, myosin phenotype, and oxidative capacity in skeletal muscle under disuse conditions, which of course includes spaceflight and Earth-based analogs. And what Boris Schenkman's lab has done really well is showing that they've shown that GSK3 inhibition in terms of spaceflight and muscle unloading can attenuate that slow to fast fiber type shift. So by inhibiting GSK3, you can preserve the myosin heavy chain 1 content, or those blue fibers. And they've done this really well using the best accepted spaceflight analog, the Heinlein suspension model. So we can do this here on Earth. Actually, Ryan Baranowski will be doing this in the next few weeks with some of the mice that we have in our lab. What you do is you suspend the mice by their tails, and so that their hindlimbs over here, they're not touching the ground and therefore unloaded. And so they're going to experience that atrophy and that slow to fast fiber type shift. But maybe what I can show you is draw your attention to the GSK3 activation. So 7 days of hindlimb suspension increases GSK3 activation and also reduces the amount of blue fibers, so myosin heavy chain 1. However, when they treated the rats with a GSK3 inhibitor, that's A, don't worry about the name, they restored GSK3 inhibition. We saw more of that GSK3 phosphorylation, and they increased the amount of myosin heavy chain 1. So they prevented that fiber type shift. So what about atrophy and weakness? Again, Ryan, who will be working with this model using a GSK3 knockdown model. So we actually have a mouse model where we've knocked down both alpha and beta isoforms. And Ryan's going to do this unloading protocol for 2 weeks, and he's going to look at muscle and bone mass. And we really like the DEXA for this because we can do a targeted approach and look at lean and bone structure in this area, in the hindlimb area. He's also going to look at fiber type composition and size, and then muscle strength and fatigue resistance. So a lot of exciting things coming from Ryan. So I'm really looking forward to it. This is the last slide, I promise. So what about calcium regulations? Calcium regulation, and again, our lab focuses on the Scintica pumps. And I'm proud to say that this was recently published, actually accepted for publication on Monday. This is Jessica Braun's side project, in addition to her PhD work. We got it accepted on Monday, so it's in press. And what she's showing is that in male soleus and female soleus, it's really important that we look at the effect of biological sex. We find that circuit function is impaired. So again, looking over time, measuring free calcium, when you provide ATP, the circuit pumps can use that to bring calcium into the SR, reducing the amount in the cytosol. You see that really nicely in both GC, ground control, and vivarium control. So those are two controls that are on Earth. However, when you look at the flight curve, you can see that when you provide ATP, there's really no circuit function that's occurring. So one of the things that we are looking forward to doing is testing whether or not GSK3 inhibition can restore circuit function. And that could set these muscles on a good path in terms of atrophy and weakness. Anyway, I've probably taken too much time talking. So I'll leave it there. Say thank you to my students, my lab, Scintica Instrumentation, and thank you also for this platform, my funding, and NASA for sharing the samples. And I'll take any questions.
Thanks, Val, so much for the great presentation. And as we have reached the top of the hour, there were a couple questions that came through, and they are actually quite scientific, Val. So what I may do is actually put you in touch with that person or grab the answers from you and pass them on.
Sure.
So at this point, I would like to thank everyone for their attention. Val, of course, for the great presentation today. I hope that all of you found it very relevant to the work that you're doing back at your labs. And if in the days and weeks to come, you have questions about the science behind what Val was doing, the systems that he was using from us here at Scintica, which was the Insight DEXA and the Prospect T1 ultrasound system, I encourage you to reach out. And again, thank you so much for your time and participating in our event. And we hope to see you at a future Scintica instrumentation event. Take care and have a great day.
Thank you.